The overall goal of the following experiment is to create an ex vivo model that replicates normal and abnormal hematopoiesis. This is achieved by fabricating a scaffold made of polyurethane using the thermally induced phase separation technique, or TIPS, which produces a highly porous, non-biodegradable scaffold in the form of a petri dish. Next, the scaffold is cut into cubes and is then coated with extracellular matrix proteins to aid cellular adherence when seeding the cells. The scaffolds are sterilized using UV and ethanol, and they are incubated in media until seeded. The next step is to isolate mononuclear cells from cord blood samples, which are used to seed the scaffolds. Results have shown that this model supports the growth of human cord blood mononuclear cells and primary leukemic cells in the absence of exogenous cytokines, based on data from MTS, SEM, and flow cytometry assays.
This method can help to answer key issues in the hematology field, such as creating an experimental model of the bone marrow to study cell interactions and also to explore novel treatments in leukemia.
Though this method can provide an ex vivo biomimicry of the bone marrow, it can also be applied to other systems, such as neuronal cells, for the in vitro reconstruction of the neuronal network, as a model to understand the natural neuronal outgrowing and also to study neurological disease.
The implications of this technique extend towards therapy of blood-related disorders such as leukemia, because different drugs can be tested towards their treatment in an environment similar to the human body.
To fabricate PU scaffolds,